let's get started so everyone can get back to their lunch and their day, as well as Alistair. Thanks so much for putting your hand up to help out with our seminar series today, Alistair. He was just saying he's not a member, so we might have to work on that. Perhaps this is a good way to get people to join. So Alistair is our April presenter for this month, and his title is very enticing, as always with Alistair: sex and drugs. So Alistair studies the evolution of fungi, including rust, smuts, fusarium, and now he's moving on to magic mushrooms. He's completed his postdoctoral fellowships in the US, Europe, and South Africa, and he's currently a research fellow at the University of Queensland, where he's doing this very interesting project that I'm sure is going to get a lot of attention. So Alistair, off you go.
Bless. Thank you, Jordan. Yeah, I didn't volunteer. You did ask me. And I'm always happy to speak. Just try and shut me up. Well, if you don't know me, I'm Alistair. I've worked on fungi for 15 years, and I've always worked on them as the bad guy. And what a refreshing change to work on fungi as beneficial organisms instead of as pathogens. And my foray into working on mushrooms is with psilocybin, or psilocybe, I should say, and they produce psilocybin, and everyone's talking about it. In fact, if you don't know what psilocybin is, then you really need to read a paper. I can't keep track of all of the developments in psilocybin research. It just feels like weekly or monthly there are new clinical trials that are finishing up. The one that really sticks with me from either this year or late last year is that a study that was looking at the treatment of people with depression using psilocybin. I think a third of them were in remission for depression 6 months after one dose of psilocybin. And that is just phenomenal. Like, there's so much potential around psilocybin, and it's why everyone around the water cooler is talking about it. And because humans are interested in humans, we're the big model organism, we haven't even begun to start looking at how psilocybin can be used beyond humans. So in cases of animal welfare, for example. So that's what this last little point is down here. And sorry that I haven't read any of this. That's just a distraction, my bad. Since I started working on psilocybe, which was in July in last year, a little piece came out in the ABC, and people started contacting me, people of this big community. And I thought, well, I better pay attention to who's contacting me. And I've kind of summarized the community on this slide. And I've probably received, in the hundreds of emails from people throughout this network. Most of the people contacting me are those who are interested in psilocybin for clinical use. So I get at least 5 emails a week, if not more, usually with a horrific story explaining anxiety, addiction, or depression, and how they are a candidate to be included in my trials on psilocybin. Of course, I'm not running any trials on psilocybin, so I kind of have a form letter where I say, hey, I work on the evolution of these mushrooms, not on the clinical use. And what's the other kind of people who it's been really interesting to meet are the people who want to start producing psilocybin. And that's probably because of that price tag of $7,000 per gram for psilocybin in clinical use. And I've got friends who are mushroom growers, and they were really excited about, they've been practicing growing mushrooms for so long. They have the expertise, they've got the infrastructure, they're ready to go to start producing psilocybe. But then after that first news release, I was contacted by a lot of cannabis farmers within Australia and Canada and the United States, all interested in kind of branching out into production of psilocybe or psilocybin. And I kind of said to my buddies in the mushroom industry, well, I don't think you guys are going to compete against them because they've got the capital, they've got the experience dealing with scheduled substances, and I think they're going to be the first off the mark. In fact, they already are in Queensland. The cannabis growers are the first ones with the licenses to start producing psilocybin. And just one little anecdote, one of the first cannabis growers who asked me about growing mushrooms said, how do I get the seeds for these things into Australia. So I think that says it all. Then, of course, this is my favorite part of the community. It's the part that I'm in, the mycology community, which is everyone who likes every kind of mushroom, no matter what. And then a special part of the mycology community are those who are really interested in psilocybe and magic mushrooms. And there is so much knowledge in Australia of people who are interested in psilocybe. In the last couple of weeks, I've had someone who lives in South Australia, and he's been collecting Psilocybe subaeruginosa from around Australia, and says that the mulch where he collects it maintains the phenotype of the mushroom every season. So there's just all this knowledge and experience within the community. It's just not necessarily in the academic community. And yeah, I just love tapping into this knowledge, and people have been sending me spores, and it's just been such a delightful community to join. And joining it was as simple as getting a permit to have psilocybin. So easy, so easy. They don't run any background checks. Anyone can get one of these. I'm just joking. Sorry, it didn't come across really well. What they were really confused about is that I'm not running clinical trials. So they were saying, how much psilocybin are you going to have, Alistair? I said, I don't know. And so you'll see down in this little strength section, they've just put, well, various. He doesn't know. I can have 150 mushrooms at any point in time and 500 active cultures. And I prefer to have 0 mushrooms. So every time I deal with a mushroom, it's just a nuisance because I have to dry it and then put it into the herbarium. And at our herbarium, we like dried leaf material and cultures. Not that we don't put mushrooms in there. So really I deal with cultures most of the time. And I just try and throughput them into the herbarium after I've either studied their mating or extracted DNA for a genome sequence. And I was just talking with Jordan about how much more progressive the Queensland legislation is than the New South Wales legislation. We were just shooting the breeze before. In Queensland, they determine how much psilocybin you have and then apply a penalty. In New South Wales, they weigh whatever you've got, whether it's wet or dry, and then they extrapolate how much psilocybin you have, and then they throw the book at you. So surely this is actually a slide from an old undergrad talk that I was giving about psilocybe, and I probably don't need to do this for the AMS community, but why not? Because it's so much fun to talk about psilocybe. So psilocybe is now in the Hymenogasteraceae. It's a monophyletic group. And within the agaricales, those spore prints are just so informative. And so everything in psilocybe has that purpley brown spore print, and you can see this guy's getting quite the shower underneath there. The other thing about psilocybe, in this monophyletic group, synapomorphy, is that they produce a secondary metabolite by metabolizing tryptophan through a pathway of 4 to 5 genes that produces a tryptamine, psilocybin, that compound that we're all so excited about. And that's this blue stain here. So a lot of psilocybe, well, all psilocybe technically, except for one, produce psilocybin. They all will have a purpley brown spore print. And so all except one will have that little blue stain down the bottom. So their sister genus in the Hymenogasteraceae is this little group of mushrooms up here, Galerina. And I just find it so amazing that they share the most recent common ancestor, and psilocybe has this secondary metabolite, psilocybin, that's hallucinogenic, whereas Galerina has a secondary metabolite, an amatoxin, that can destroy your liver and cause a painful death. Then the last mushroom pictured up here is Paniolus. How embarrassing, I can't remember its family at the moment. All these things occupy the same ecological niche, so as saprotrophs degrading wood or dung. And it is a strong hypothesis, very well supported, that there was a horizontal gene transfer of the cluster of 4 to 5 genes that produce psilocybin from psilocybe into Paniolus. And so Paniolus, there are 5 taxa in that group that produce psilocybin, and the rest do not. And just that it's a horizontal gene transfer event is supported just because you've got movement of an entire cluster of genes rather than a single gene or whatever. So it's often a signature of horizontal gene transfer, which is also what I work on in Fusarium, if you're ever interested in listening to me speak about that. So yeah, that's just a breakdown of everything I've just said. Let's forget I even put it up. So since I started in July last year, really, I've only had an opportunity to collect Psilocybe cubensis, and photograph them. And here's just a couple of photos of cubes. You've got that nice annulus that starts to stain purple and has this black look after the pileus opens up. There's a little bit of that blue stain. And of course, in that dung habitat, there's that purple spore print for your people who like to see how to identify them. Here's a black spore print. So among these purple spore prints, you've got 2 black spore prints, and those are paniolus. And then the spores down a microscope, this spore here is 15 microns. So the lovely, pretty images aside. Now, also with AMS, I'm sure I don't need to break down a life cycle of a mushroom, but because I've worked in Basidiomycota for 15 years on rusts and smuts, and now Agaricomycotina, so the 3 subphyla of Basidiomycota essentially, I'd still just like to talk about it a little bit, because everything I do is based on this life cycle and the haploid stage. So within Basidiomycota, what they really share in common is that when they're growing as a mycelium or a hypha, they have two nuclei, so 2 haploid nuclei, and this is just a dikaryotic growth. In mushrooms, in Agaricomycotina, that nuclear fidelity is maintained by clamp connections. So as the hypha grows, you'll have one of the mitotic divisions of a nucleus will go with that, and a clamp connection will be produced to send the other nucleus after a mitotic division through it. When you don't have clamp connections, such as in rusts and smuts, you can get all sorts of funky arrangements of nuclei, and it's unsure of how nuclear fidelity is maintained. Even in mushrooms that don't produce clamp connections, you'll get some really cool kind of genetic stuff going on with the sharing of nuclei. So bless those clamp connections in Psilocybe for maintaining that nuclear fidelity, because that makes our lives easier. When you've got that fungal hymenium produced of dikaryotic mycelium and the conditions are right and everything's right and the mood is set, it's time for sexual reproduction and the fruiting of a mushroom, in which you have that karyogamy, so those 2 dikaryotic nuclei join to produce a diploid nucleus, and then you can have meiosis, so recombination. And those haploid spores that are produced with a new recombinant nucleus, their main goal is to meet up and become a dikaryon again. And in smuts, you'll find that haploid stage is non-pathogenic growth. It doesn't become a pathogen again until it gets its buddy and becomes a dikaryon. In rusts, the haploid infections like spermagonia, they're really mild, minor, forgettable. You walk right past them. And with mushrooms like Psilocybe, you'll see photos soon. When you're growing a haploid, it just doesn't look as robust. You know, it's got one thing on its mind, becoming a dikaryon. So I haven't tested, and I probably won't test whether psilocybin is even produced in these haploid cultures. But I work on haploids because they're the easiest for genetics. And in the future, technically, with all the haploid cultures that we have, we can cross them with compatible mating, compatible mating types, which I'll cover toward the end of the talk with some new knowledge. So this is kind of where I am with my project, or with what I'm doing. I shouldn't even call it a project. Nothing's funded. This is all just done out of my interest. I have 4 days a week research covered by UQ, in which I do many things. Most of them are on grants. This one's just kind of on the side, a special interest project, which is so cool to have the freedom to work on it though. So I really have targeted the project to 2 mushrooms in Australia, 2 species of Psilocybe, subaeruginosa, which is most likely an endemic. So this mushroom was described in 1927 by John Burton Cleland, who is just an absolute hero. He described it from South Australia, from New South Wales and Victoria, from grasslands, from degrading woody material like eucalyptus debris, and really from natural native areas. He described that in 1927, and then almost 20 years later, so in 1946, a really similar looking mushroom was described from the UK called Psilocybe cyanescens. And as anyone who works on mycology in Australia knows, we normally take the names from the Northern Hemisphere and apply them to our mushrooms, whether they're conspecific or not. It's just an artifact of us lagging behind the knowledge in the Northern Hemisphere. So it's really interesting that a mushroom was described, a near identical looking mushroom was described from Australia before it was described from the Northern Hemisphere, from Kew Botanic Gardens, a weedy location. So Psilocybe cyanescens spread through Europe, and it was found all up and down the west coast of the United States, and it became one of the go-to mushrooms for recreational use, and people started throwing patents on it for extraction of psilocybin and even dosing with Psilocybe cyanescens, not subaeruginosa. And so if this mushroom is endemic and Australia is the center of origin, then there are impacts for biopiracy, for use under the Biodiscovery Act, which is in Queensland, and the Nagoya Protocol for people being able to commercialize these mushrooms in the Northern Hemisphere. So I think it's pretty, it would be nice to show that it's native to Australia and that we have the center of origin of this particular mushroom. On the other hand, we've got Psilocybe cubensis, which is kind of like the lantana of magic mushrooms in Australia. If you go to a cow farm in Queensland, not only do you see a lot of lantana, but you also get to see a lot of Psilocybe cubensis growing out of manure. And just reading the old literature from Queensland in the 70s, when the UK mycologists were coming here, they talk about how prolific cubensis was just around the Brisbane area, which I just love reading those old stories. They're so fascinating. So the center of origin, Cuba, cubensis, described from Cuba, and talking with the community here, such as people like Kane, hi Kane, the center of origin of Psilocybe cubensis, I think, has a bit of a question mark over it. It is an associate of mammal dung. So somewhere where there's some big mammals might be a good clue. It's probably not from Cuba. It will be really interesting to look further at where that came from. In Australia, it'll be interesting to see how it was introduced. We're going to see some work that I did on the weekend specifically for this talk to look at where it might have come from, and to also look at how many times it might have been introduced to Australia. So I just want to break down all those points just so that you've got them in writing. You can also get all of this information off my blog, which I'll put up at the end. So subs described in 1927, 20 years later, a Northern Hemisphere lookalike described from Kew Botanic Gardens in a weedy location, and it doesn't occur in natural ecosystems in the Northern Hemisphere. And this is well before even the effects of psilocybin are kind of known in the Western community. And I'd love to talk later about psilocybin use before Australia was more colonized. So it's kind of such an interesting topic, and I fear that we've lost a lot of knowledge about indigenous use of magic mushrooms. But it's also a sensitive topic to talk about. Yeah, Psilocybe cubensis is prolific on the East Coast, and both of these mushrooms are under study for commercial use. And it would be cool to know their centers of origin, where the genetic diversity is, and that will help inform all sorts of policy and legislation about who profits and who benefits from these things. And you can read those research questions on my blog. Feel like we've covered that. So a little bit about my methods. If I collect a mushroom, and I have a mushroom which is in the rare occasions that I do, if it's a really heavy blue stainer, I'll sometimes keep that genotype by surface sterilizing the stem and then just culturing directly from the stem. But that's on the rare occasion. I don't have very many diploid or dikaryotic cultures of psilocybe. Most of mine are haploid. So I do a 16 streak from a spore print, and then isolate 5 or 6 haploid cultures from each mushroom that has been sent to me for a spore print. And then I grow that up in liquid for the best DNA extraction for genome sequencing. And so right now, since starting the project in July, I have about 56 haploid cultures of Psilocybe cubensis. I've got about 10 Psilocybe subaeruginosa. One of those is a dikaryon collected by Nigel Fechner. Thank you, Nigel. In fact, it was the very first one that was collected in the project. We've sequenced 7 genomes of subs and 3 genomes so far of cubensis, but recently I've just started growing up. I've got money somehow, it came from the sky. I've got 32 genomes ready to go for sequencing of cubensis. And this is what happens when you take a look at those genomes a little bit more closely. This is just kind of to summarize what they look like. This number in the middle is the number of contigs. The smaller that number gets, the closer it approaches the number of chromosomes. So we want that number to be really small, even though this number, which is the genome size, so this particular genome, 48 million base pairs, 48 meg, is made up of 2,477 contigs. So this one on the end just looks so funky. And that's because it's the dikaryon that Nigel gave me. So it's got a larger genome size and a lot more contigs. And the reason is because it's a dikaryon. So it's just so much harder to assemble because you've got 2 nuclei in there, and it's really just annoying to work with dikaryons in terms of when you've got the opportunity to work on haploids. So yeah. When I take those genomes, I just to make sure that I've made sure that they're haploids just by that assembly. And then just to make sure what they are, I just whack them in the tree, pull out the ITS and pop it into the data from the community. And so in this cyanescence complex, we're interested in this kind of teal deal here and this green one, which is cubensis and subaeruginosa, those 2 complexes of taxa. So these are all the names that have been applied in the subaeruginosa species complex. And when I see something like this, which is just all the ITS sequences grouped, excluding non-parsimony informative characters, this is kind of an indication to someone like me that we need to get in there with a population approach rather than maybe a multi-locus gene approach. And that's also the same with this. This is just another way of visualizing it. This is a splits tree network where you see every relationship among the data rather than a dichotomous hypothesis of evolution in a phylogenetic tree. So whenever I can, I encourage people to learn how to read these. So here's our subs and our cubes. The more reticulation you see, usually it's a sign of recombination. In this case, because we're only looking at the ITS region, it can also be a sign of convergent evolution. And when you've got a couple of genes in there, it can be incomplete lineage sorting. So yeah, in this case, it's just every hypothesis of evolution that you can have among the data. But the message is that this is a population question. So this is old news as well. This was recently published in the ABC. I don't know if it was published, it was reported in the ABC. I was lecturing in one of the mycology classes available at the University of Queensland, and a student asked, why does Psilocybe cubensis only occur on cow farms? And with confidence that I'm not entitled to, I said, I'm sure it would occur everywhere. It's just that that's the niche. It produces spores that blow around, like huge amounts of spores. I'm sure it's everywhere as a saprotroph, wherever it can get carbohydrates. We just see it fruit because it's got that niche of dung. And so when I got home, I was like, okay, I think I can test this a little bit more because we've got this Bioplatforms Australia. We've got the soil sequencing database where they've taken soil samples from over 3,000 locations across Australia, mostly from natural or national parks, natural areas, national parks. And then they've done ITS sequencing of the fungi in each soil sample. And my hypothesis was that we were going to see Psilocybe cubensis everywhere. And I was wrong. Rather, what we see is that Psilocybe cubensis was not in any of those 3,000 soil samples, more than 3,000 soil samples sampled from native areas. So we see these 4. So the ones that are in bold are actually the sequences from base. So here's our subaeruginosa. This is actually also a little bit of a window into that. Just because something isn't in here doesn't mean that it doesn't occur there, because there was definitely sampling sites in South Australia, New South Wales and Victoria, but subaeruginosa was not, there were no sequences of subaeruginosa in those sample sites. There were from WA and Tassie, where it's really prolific. Kane taught me what this one was. I think I've forgotten. Oh, that might be Paliculosa. Oh, I can't remember. We can ask Kane later. And then Papuana down here. but the exciting one for me was the sequences from Kakadu and Litchfield National Parks up in the Northern Territory. So represented by a couple of hundred sequences, which is super interesting. And because we know that this particular ITS type clusters with Psilocybe, and we know that Psilocybe is a monophyletic group of fungi that produce psilocybin, we can be pretty sure that this is a hallucinogenic mushroom. And so tentatively, I've applied the name Psilocybe brunniocistidiata. And the reason is because this godfather of Psilocybe, Gaston Guzman, when he was describing mushrooms from PNG, from those subtropical to tropical rainforests, he highly suspected that this mushroom was going to be sister to Psilocybe youngensis, described from Bolivia. And to see this relationship in the tree of the mushroom from the Northern Territory being sister to youngensis, so in the position that Gaston Guzman had hypothesized for brunniocistidiata, and just that proximity of Northern Australia to PNG, I thought in the meantime, considering we don't have a sample or know what this thing looks like, we could possibly speculate that it's going to look something like Psilocybe brunniocistidiata. Whether I'm right or wrong, we'll have to wait until we have a physical sample in hand. So the next question that I was just recently asking with publicly available data is Psilocybe cubensis introduced to Australia via the recreational community? And recently, like in the last couple of weeks, these people, Psilocybe, have released genomes of almost every cultivated line of Psilocybe cubensis. So they've made them publicly available. The jam is that they haven't sequenced haploids. Rather, they've sequenced the spore print that these companies who are selling the mushrooms send out to people. So from that spore print, you can imagine each haploid spore has a different genome than its sister spores. So they're all recombinant offspring. So assembling genomes from these spore prints, which would contain millions of different genotypes or haplotypes, I should say, is going to be a little bit of a challenge. So what I did is just to kind of get away from all of the conflicting, I don't care about synteny or where something is. I'm trying to still somehow encompass the allelic variation and steer clear of the mess. So what I did was just annotate with a hypothesis what all the genes in those genomes were, in those 80 or so genomes. And then I only looked in the nucleotides that were responsible for coding genes. And you're about to see what that produces. So this is about 85 genomes of Psilocybe cubensis that are cultivated commercially. And down here in this bottom right-hand corner, the 3 Psilocybe cubensis that I sequenced genomes for. And I can't wait to see where the rest of those 32 are. So again, with these splits trees, you've got reticulation, meaning recombination. What I haven't spoken about yet is branch length. So branch length, in this case, this is made from more than 500,000 SNPs. So 500,000 nucleotide differences among that protein coding sequence data, or sorry, I should say nucleotide sequence data that encodes for proteins in the genome or encodes for genes in the genome. So branch length is informative of genetic difference. And when you start to see that things branch lower down or split away from each other lower down, it just means that they're more distantly related. So it's genetic distance. So because you're looking at every hypothesis of evolution completely. So now you really branch length is showing distance. So across 500,000 nucleotides, more than. So just based on distance, you can see that in the 3 genomes, which are from one population on the Gold Coast in Southeast Queensland, there is more genetic diversity in those 3 genomes than there almost is in this whole golden teacher little cluster up here. This is P envy. So all of the big names are in here. So what this tells me is that in all those cultivated strains, and I just might go to the next slide. So this is just the slide of golden teacher, of that golden teacher cluster. Now there's fewer than 500,000 SNPs. So I just looked within that population. I think there's only about 200,000 SNPs in this. So within this, the way that I would interpret this, other people might have a different interpretation. you have very little recombination, so sexual reproduction going on. Even these are obligate outcrosses. I think what we have is a signature of inbreeding. So where people are crossing similar genotypes over and over again. So we don't have anything that's clonal. So the same genome hasn't been sequenced more than once, but we have these long branches, which are often a sign either of clonal reproduction, or you start to get inbreeding on those long branches when you don't have reticulation. So within that golden teacher strain, everything is really similar, and it may have all came from kind of the same parent, and people have selected for phenotypes over time, and they've ended up with this low amount of genetic diversity, even though there might be high phenotypic diversity in there. And yeah, it's just super interesting to me. These little figures here are called the index of association. As that number gets smaller, as it gets closer to 0, it's a signature of sexual reproduction. The closer it is to 1, is an indication of clonal reproduction. So we know that there are no clones in here, so because the same genome hasn't been sequenced more than once. So we don't expect this to be very close to 1. It's a little bit hard to take it, to interpret this without a little bit more knowledge of other populations. I will expect that when we look at the population of subaeruginosa in Australia, that we'll have a much lower index of association. Probably if I looked at the index of association across the whole of Psilocybe cubensis, we would also expect this number to drop a little bit, as probably just because there's a bit less linkage and a bit more signature of sexual reproduction. So yeah, stay tuned. I'll update you on the blog about all of this. The next little thing I want to talk to you about, speaking of all this inbreeding, I want to talk about sexual reproduction. So when you have 2 haploids, this is Psilocybe subaeruginosa again, when you have 2 haploids that want to meet up, they change from that haploid flimsy, weak, boring growth, and they become a little bit more robust and start growing this white fluffiness. When you have something that's incompatible, then you've got this no means no zone where there's nothing going on. They do not want a bar of each other. And so I think that Perry spoke to you a little bit about sexual reproduction in Basidiomycota at your last AMS meeting. Aren't you lucky? I'll touch on this real quick, just in case any of you missed out. Essentially, mating in fungi or mating in Basidiomycota is controlled. Hold on, let me go back a notch. Let's go back to something we all know. Humans, XY chromosomes is a little bit similar to Ascomycota mating, where you've got often just compatible mat loci that are mat1, mat11 or mat12, and that you can have a sexual cross. In Basidiomycota, we add another locus, another locus that regulates mate compatibility. So we've got a homeodomain locus, and this locus is usually really, really variable. The more variation we see in it is often, I'm going to use this locus actually as an indication of whether Psilocybe subaeruginosa is native in Australia. I expect to see a lot of variation across the entire population across Australia at this HD locus. So we expect a lot of variation. At that locus, there are 2 genes. In all of these photos, it's just these 2 little green things pointing in opposite directions, HD1 and HD2. So at the homeodomain locus. So 2 genes that must differ to whatever they're crossing with for a compatible cross. Then a pheromone receptor locus, which is a pheromone or a receptor gene that is involved with mate recognition. Now, whether those genes are linked or whether they're separate in the genome will indicate whether they're bipolar or tetrapolar. If they're really, really close in proximity to each other, they're probably linked to each other genetically, so they'll move in recombination, and that's called bipolar. So it's like having only one locus that controls mating, even though you've got all these genes, you'd still call it just one locus. When you've got a separate pheromone gene to a HD gene, so on different chromosomes, for example, then you've got something called tetrapolar mating. So you need to differ at 2 sites to have a compatible cross. So it's almost like having 4 different genders in a cross. You can end up with having a, because this pheromone receptor locus, there's less variation than the homeodomain locus. So sometimes over time in some fungi like Coprinellus, formerly Coprinus, you've had a switch from tetrapolar mating back to bipolar as the pheromone receptor locus just kind of becomes redundant. Like it just doesn't have any variation at all. It no longer controls mate compatibility, even though it still may control mate recognition. So let's take a look at how those genes look in the population of Psilocybe subaeruginosa with genome sequences. So this is the HD locus, and what's pretty interesting, so here's Nigel's one is 01, and then the population that I collected from a rainforest on the range in Southeast Queensland is 02. So I've got 4, 5, 7, 2, and 6 had one genotype of HD2, and number 8 had a different genotype of HD2. And that's exactly the same with HD1. So we're pretty confident that HD1 and HD2 are linked. And I'm just chucking this up. This is interesting. This is HD2 in every single Psilocybe cubensis from that genome data. And what's amazing is that there is very, very little genetic diversity at that homeodomain locus in the cultivated lines of Psilocybe cubensis. So super interesting to me. It's going to be interesting to see what can cross within the cultivation community. So then at our pheromone receptor locus, we've got 3 of them in subaeruginosa, and we've got 1, 2, 3, 4 in Psilocybe cubensis in those 3 genomes. So this was a little bit unexpected to me, though not totally unexpected. What's interesting is that in the grand scheme of Basidiomycota, mycota, it's not unexpected to have several pheromone receptor loci. And often they'll be linked because one will control the pheromone and one will control the receptor. Let's not get into that. So these are segregating in that little population that I sequenced. So there's 2 alleles and for each of those 3 pheromone receptor genes. So I thought, well, cool. I know the homeodomain genes. I know which of those will cross, and I know the pheromone receptor genes. So a little quick breakdown is we've got an allele for HD1 and an allele for HD2. So remember this, 8 was different. And then our pheromone receptor genes, we've got 1, 2, 3 of them, each with 2 different alleles. So using the knowledge that opposites attract, pretty much 8 is the one that's going to be crossing with everything else. And then based on these pheromone receptor loci, which also technically need to differ, the only thing that 8 should cross with is 5 and 6 if all of these pheromone receptor genes are involved in mate recognition, in mate compatibility. So I was expecting, you're about to find out how wrong I can be. I was expecting 5 and 6 to cross with 8. Let's have a little look at what happened. And I only found this out yesterday. So I did this experiment just for this talk. So here's 5 times 6. There's that no-go zone. It's not on. But then here's 8 times 6, which was a real letdown watching this not cross over the last 2 weeks. And so everything that I tried to cross unexpectedly did not cross. So not a single dikaryon was formed. And unfortunately, I have no idea which isolates I photographed in that compatible cross. So probably in the next 2 or 3 weeks, I'm going to have to set up quite a large crossing experiment, which will have to happen when I have time. So yeah, that is less exciting. No crosses. I would just like to thank all these people who have been sending me spores and as part of the study and brainstorming ideas with me. I'm always happy to receive spore prints. If I do receive mushrooms, then there's not much I can do with them. I just put them into the herbarium. And of course, because psilocybin is a scheduled substance, you're collecting it for me at your own risk, and you're not covered by my permit, which, you know, it's so sad that people can be penalized for picking a mushroom, but that's just the way it is. So yeah, these are kind of like the messages that about my project, which I don't even feel like going through at the moment, but you can read them if you want to. And really everything that I do is I update it all on this blog. You can find it through my UQ researcher profile if you are really interested. So thank you.
So much, Alistair. If anyone has any questions or thoughts, pop them in the chat. How many cultivated strains are there, and like how long have we been, them, commercially cultivating? We as humans have been commercially, cultivating these.
Yeah, well, I guess after 1958, right, is that kind of point where people, like the other community, so in Central America and South America, where psilocybin use has been known for a long time, I don't even think it was, I don't think that they were cultivated. I probably, my fingers off the pulse, we'd have to talk to an ethnobotanist, but because it was a sacred journey and ritual and seasonal, I don't think they cultivated it. Whereas, yeah, cultivation, I think, has been going on for probably, 70 years or so of Psilocybe cubensis. I would guess, and I guess
and there's like hundreds of strains. Yeah,
mostly inbred from that analysis.
Joanna has a question. Fascinating. So what would be your alternative hypothesis on the mating compatibility? Now, would you think there's another locus involved aside from the two?
Yeah, I do, Joanna. Good question. Because of that HD, the lack of variability at the homeodomain locus across cubes, and we know that they cross, something is up. And I only found this out yesterday, even though I'd seen it coming. So I've sent it to people who know more about mating than me. I am not sure, but that homeodomain locus, because I'm really going to rely on it for a lot about variability, and I expect a lot of variability at that locus. I'm really hoping it's going to be the one that is telling us all, oh man. I'm going to share my screen again so I don't have to look at myself.
Sorry, I made you big. You can go tiny again. Cool. And you're just sort of doing this, well, not in your, well, in sort of your spare time. But you did say that you've gotten some support and funding for it.
No.
From a man, you said.
From what? From these people who are sending me scores.
Contributing, yeah.
And like, obviously this couldn't happen without the community, because without me being able, like, I don't have any funding to get in the car and even go and collect.
No, yeah.
So without the community helping, and at their own risk, which is even more amazing, like it just shows how much people care about this issue. It's all unfunded. It's all funded creatively through grants that are supposed to be on plant pathology.
Yeah, I thought it was great. Also, sort of a nice highlighting the thinking about your project before you do it with that, the encyclopedia of genomes, where obviously it would have been more helpful if they had gone the effort at the beginning to get their haploid cultures before they went and
I think that their beds were made. I have a feeling that they're not allowed to, like, they really wanted to do it above board, so they didn't want to start producing new culture. So that was their body.
So so research has been limited by the legalities in the past, possibly. Right.
Yeah.
Oh, okay. Well, I think unless you had anything more to add, Alice.
Questions. How do I
they're in the Q&A.
The subaeruginosa spread out of Australia? Oh, hey David, how's it going? Well, just where cyanescence was first described in Kew, just imagine someone digging up a plant to take to the botanic gardens over there. I mean, Brett, Summerall, Jordan, and Tom, so 3 curators of herbaria, have all told me about Psilocybe subaeruginosa growing in their gardens at those collections. So someone moving a plant, and this is called, it's a saprotrophic invasion essentially, which is becoming a hotter topic, I think. So just the spread of saprotrophs around the globe. They've been underlooked for pathogens. And then, have I integrated any New Zealand strains labeled subs in my phylogeny? Well, in the phylogeny, there's a couple of Werrowa, maybe there's a Aucklandii, so whatever was publicly available. But just because, again, I want to make sure that I do this where I've got permission to do it, I think as soon as I start to include New Zealand specimens, it becomes, I think they become like a couple of issues. So I really, I'll leave that for the New Zealanders. So Werrowa just sits in that subaeruginosa clade. I think Macarore is also described from New Zealand. So yeah, really going to be a part of that subcomplex. And then, yeah, Bevan, hey, all right, cool. Grouse, I think I'm done. Sorry, I felt so lackluster. I needed to have a Barocca or something.
Cool. Great, thanks so much, Alistair. And thank you everyone for joining today. It will auto prompt you with a poll. Please take some time to fill it in. Thanks so much. See you all next time. Bye.